Okay, our next speaker is Liang Huang, who is going to talk about optimized mRNA design.
Thank you. Thank you. Thank you, everybody. Thank you for the organizers for inviting me. I think the previous talk ended by saying if you can enlarge the half-life of mRNA, you get more proteins. That's exactly the topic of this talk. And my background is quite different from everybody else's. I'm a computer scientist. I'm an artificial intelligence researcher. And my area is computational linguistics. But in this talk, I'm trying to convince you that linguistics and biology are the two sides of the same coin. And actually, the new COVID-era problem of mRNA design can be reduced to a classical problem in my field, computational linguistics, which we solved in 1961, 60 years before the pandemic. So it's a very exciting story that we published in Nature this year. And let me try to, okay, so I don't need to give this background because we all know about this. And mRNA molecules are rather fragile and they tend to degrade. So if they degrade, they got less proteins translated, and in turn, you got less antibodies induced. So my question is, how do you design more stable and efficient mRNA sequences computationally using an algorithm? So here's the mathematical problem. We all know that translation is deterministic, but design is the inverse problem, which is non-deterministic because of the redundancy in our genetic code. And like Valium has 4 triplet codons, and leucine has 6, and so on and so forth. I think that one of the tutorials in this morning, Dr. Eckhart, you mentioned this as well. So if you multiply the number of choices together, you got a humongous number, and that number is coded as well this morning, 10 to the power 600 something, exponentially many. If you want to try them 1 second a sequence, enumerate them, you take billions and billions and billions of years. You wouldn't be able to finish before the universe dies. So how do you search for the best one? Well, Moderna's paper in PNAS right before the pandemic, December 2019, suggested that you want to find the most stable mRNA, which stays longer, the longest in your cell, which produces more proteins. But the question really is how? How do you find that most stable mRNA? So here is what we got from AI, from my original field. And we borrowed 2 concepts from my field. The first is word lattice, which is just a representation. You just pack the different choices of codons into a little graph called lattice with ACGUs on the edges. Like this is the 4 choices of Valium, this is 6 choices of leucine, and so on and so forth. And then you stitch all these little mRNA lattices together. You got a very long, very thin mRNA lattice, which is very nice in the sense that from the beginning to the end, you got each path corresponds to a mRNA candidate sequence for the COVID-19 vaccine, and there are 10 to the power 600 something path in it. But that doesn't really solve the problem, because the problem is, how do you find the most stable path in this representation? So here comes the second, or the most crucial idea in this work, that is borrowed something called lattice parsing, a very powerful technique from AI. So as you hear me speak right now, you're doing speech recognition in your brain, even though you're not recognizing it. But because I'm not a native speaker, I have an accent, I speak fast, and there's probably background noise, you couldn't hear clearly whether I'm speaking veal or meal in isolation of context. But with sentence context, use language models, you can know, oh, you know what, I like this meal is more likely than others. It's more grammatical. So this is the concept of lattice parsing, which is a very efficient algorithm that you give the lattice, and then you give an English grammar, which encodes like grammaticality, and then you can very efficiently parse all the sentences in this lattice without enumerating them one by one, because that would take too long, and quickly find the most likely sentence, most grammatical, most plausible sentence, and its syntactic structure. I like this meal. So we just borrowed the same idea. Oh, by the way, this was really done in 1961, and there's a reference for that, which is in German, which I don't understand, but that's the classical algorithm. And we just took the same algorithm. We just changed the input and output. So we just replaced the word lattice by the mRNA lattice, which encodes all the design space of this COVID mRNA vaccine. And we just replaced the language grammar, the English grammar, by an RNA folding grammar, which encodes stability or free energy in biophysics instead of grammaticality. And run the same algorithm. I mean, the algorithm itself doesn't change, just the ingredients change. And that algorithm will fold all the mRNA sequences in this lattice very efficiently without enumeration. And in just about 10 minutes, you can find the most stable mRNA sequence and its optimal secondary structure. So here is the optimal secondary structure. And on the left, you have the unoptimized wild-type sequence, which you can see is very unstable because it has lots of single-stranded regions, very easily degraded. But just after 11 minutes of AI optimization, we found a very different sequence, completely different sequence with a completely different structure. And that structure is mostly double-stranded. In a way, we kind of turn RNA into a DNA. It's mostly double-stranded, so it's much harder to be degraded. And we can prove mathematically that this is actually the most stable sequence with the lowest free energy among all the 10 to the power 600 some alternatives. So all of this story happened very fast in the first few months of 2020 because everybody was working so hard on COVID. So we started on January and we put this preprint online in April. And a lot of mRNA companies noticed it. One of them, Seminar RNA Therapeutics in Shanghai, China, which is a leading mRNA company in China, volunteered to do the experiments for us, and their experiments are quite surprising. They show that our vaccines can lead to up to more than 100-fold increase in antibody response over the standard code optimization techniques used by everybody in this audience, like BioNTech, Moderna, and CureVac. And it will also significantly better than BioNTech sequence in a head-to-head study, and also some other vaccine as well. So here's some more technical details. So we talked about stability, but actually that's only one of the two dimensions that we optimize. It's actually a joint optimization of two dimensions. Stability on the x-axis, the lower the better, the more negative the free energy, the better in physics. But there's also y-axis, which is the translation efficiency or code optimality, right? You want to use, as we all know, the frequent codons preferred by the human genome, and want to avoid rare codons so that you can translate faster. But that's the original way of standard way of doing sequence design. That's code optimization. But that's very easy because that's just a local change. For each codon, you prefer the optimal or frequent codons. But what we do is a global optimization because you have to correlate changes of this codon with some codon 1,000 codons away, 1,000 nucleotides away, and so on and so forth. So our work enables, for the first time, the exploration of previously unreachable, kind of very efficient, highly efficient, highly stable designs. So in a way, Moderna's paper pointed us to the right direction, but they didn't know how to get there. I mean, they kind of see there's a continent over there, but we found a way to actually get across there in 10 minutes instead of billions and billions of years. So nobody was able to cross this dotted line because that would take millions of years in energy. So we were able to go all the way to there. And here's some more experimental results from Seminar Therapeutics. So we designed 7 or 8 mRNA sequences for COVID, and they tested in vitro and in vivo. And this is gel run, and you can see indeed, our most stable sequence here was running the fastest, and H, the baseline, the code optimized baseline, which is very close to BioNTech sequence in this plot, runs the slowest. And the running speed on the gel run correlated very well with our energy calculations in theory. And the half-life, the mRNA half-life also correlated very well with our theoretical calculation. We improved half-life from 3.9 hours in code optimization to 20 hours in our design. And protein expression, we also improved a whole lot, like about 3-fold increasing protein expression. This is 48 hours in vitro. And then binding antibody and neutralizing antibody, we improved by 100-fold and 128-fold and 28-fold increase over the code optimized baselines. So this is all great and very surprising that this is without any chemical modification at all. So they were very surprised about this. And one of the when we sent it to Nature, because of course the reviewers were very picky, and one of the reviewers in Nature said, you should compare with the most widely used sequence, which is the BioNTech Pfizer sequence. By the time we started this study, the sequence was not available to us, but by the time we submitted and the review, it was made available. So the reviewer said, now you have to compare with it. But one of the other reviewers thought this is a bit too harsh because they're a product and we are research. But we still, nevermind, did a head-to-head comparison with everything else being equal. It's kind of a control variable study, just the coding sequence used BioNTech's. So indeed, on this plot, BioNTech sequence and our baseline are very, very close in terms of energy and the CAI and the code optimality. And indeed, in terms of mRNA half-life, it does very similar to our baseline, actually a little bit worse, because it's actually a little bit worse in terms of energy. But in terms of protein expression, it's actually a lot better than our baseline. But our designs, A and C, are still significantly better than the BioNTech sequence. And in terms of antibody response, our C sequence is also significantly better than the BioNTech. The BioNTech is noticeably, obviously a lot better than our baseline H, but still our design is significantly better. So the editors, being an immunologist, suggest that we might be just lucky on COVID, so you have to do another vaccine. So we just did another vaccine on varicella-zoster virus, and it showed also very good improvement. But the improvement is slightly smaller because the sequence is much shorter in this second vaccine. The COVID vaccine, because of spike being very long, is actually about 4,000 nucleotides. So the longer, the take-home message is the longer your mRNA is, the more needed you come to us using our algorithm, because the more unstable mRNA is if you get very long, right? And just to quickly summarize, so we showed an alternative way to make our mRNA vaccine work without chemical modification, that is using computational design. And this can be done it's actually complementary to and should be combined with chemical modification, which I'll show in the next few slides. And in fact, a modified version of one of our designs is actually in emergency use authorization in Laos. And this is not just about vaccines, it could be used for any therapeutic proteins or circular RNA, which I will show in a bit, and self-amplifying gene editing, anything you can come up with in this conference, you can use on my algorithm. And Sanofi already licensed it, and many other companies are in the negotiation. And actually, an independent study, which was mentioned in the previous talk by Richard Dass's group, used our design and independently confirmed our algorithm's advantages. And they published actually a bit earlier than us, before us. And this was eventually published this year in Nature, along with an official news article from Nature and a technical commentary from Nature as well. So some other side advantages of our work. So besides being very stable, we also have another advantage that because it's very compactly structured, our designs, it actually interferes less with the UTR structures. So it actually becomes more independent of the UTR choices, right? Whereas the baseline, the code-optimized sequences are not as tightly structured, so they have more freedom, more capacity to kind of interfere with the kind of important structures in the UTRs. So that means a side advantage of our work is that our work is likely effective independent of UTR choices. And indeed, in our study, we used 2 different UTR pairs, and Lepek et al. Used 3 different UTR pairs in repeating our work. They all work. And there's something new that's another preprint that is looking at circular mRNA design, and that's actually so I think circular mRNA is the next hot topic, and the talk just after this talk, the next talk will talk about circular mRNA. But linear design is a perfect fit for circular mRNA. Why? Because first of all, it doesn't use chemical modification, at least for now. And secondly, the RES and ORF interaction becomes a much bigger problem because RES is much longer than 5' or 3' UTRs. It's actually about 800 nucleotides. So linear design, as I said on the previous slide, is a very effective way of avoiding these kind of interactions because we design our ORF to be extremely tightly structured. So you can see here, linear design has only 6 pairs crossing the RES and ORF, whereas code-optimized baseline has numerous pairs crossing RES and ORF. And as a result, we can find much better circularization efficiency, stability, protein expression, and antibody response, and all those things. And finally, let me talk about chemical modification because Dr. Kariko is here. So why does my work, without using any modification at all, kind of successfully evaded, in a sense, the toll-like receptors and all these innate immune responses? I don't have an answer, but my hypothesis is that we reduced the number of accessible uridines. We actually do not reduce the number of uridines. Actually, our percentage of uridines is significantly higher than the code-optimized ones. You can see that we use way more uridines. But because our structures are more double-stranded, the number of accessible or exposed or single-stranded unpaired uridines are actually less. Actually, you can see that here C and D have only 4% accessible uridines, whereas the BioNTech sequence has more than, I think, which one is the BioNTech? I can see more than 6% accessible uridines. And also, if you compare our baseline and the BioNTech, but the BioNTech sequence has lower U percentage and substantially lower accessible U percentage. And probably that's one of the reasons why it works a lot better than our baseline. And finally, The two techniques, as I said, are complementary or orthogonal to each other because one is chemical, the other is purely computational, and they should be combined and can be combined. And there are two options that to combine them. The first is a rather trivial approach. It's just run our algorithm, which optimizes for natural unmodified RNAs, but then you transcribe with pseudouridines or mRNA pseudouridines, and that kind of works, but not taking the full advantage of our algorithm because it's optimized for a different direction. But that's actually the way one of our COVID designs entered emergency use authorization and clinical trials 1 and 2 in Laos, and there's some clinical trial data just available right now, and it's pretty good. I think I'm not part of this study. This is also from Stamina Therapeutics. But the more interesting approach would be to replace the linear design energy models from a standard energy model to a chemically modified energy model. Once it becomes available, then we call it linear design plus, and that is work in progress. It would be much better than option 1 because it would directly optimize chemically modified energy, free energy. So it should be more potential there. We have the code ready, but we haven't found collaborators we're just trying to do experimental verification of that, and haven't started on it. And we firmly believe that this option could also work because Moderna's paper, that this conclusion from Moderna's paper that stability improves translation holds with and without chemical modification. So it should totally work there, we believe. And with that, I would like to thank you for your attention. And here's our web server. You can put any protein sequence you want to design, and we will design for you. And there's the result page. You can have the secondary structures and all these things. Thank you very much. And there's also another talk from Oregon State. My colleague Gaurav will be doing a talk on delivery tomorrow. So thank you. I'll take questions.
Okay, questions? No question from, okay. I see one question here.
Hi, Liang.
Thank you.
Soon from Inceptive. Good to see you again. I had a question on the data you showed
Slides.
on.
Can I get back to the slides? Sorry, yeah, go ahead.
Oh, yeah, I had a question on the improved antibody titers.
Yeah.
You showed over the other.
Can the slides be back on? Sure.
Oh.
Sure. The COVID results?
Yeah. I noticed on the bottom of the slide that the S2P mutation had been reverted to the original.
Right. Yeah.
And also,
because when we started this, we started this very early, so the 2P mutations were not known at that time back in April of 2020. So the initial sequences that we designed for the protein sequences did not have the 2P mutations. So that's why we had to kind of to do a head-to-head comparison. We have to basically reverse the 2P mutations to use the original spike sequence.
But from my understanding of the antigen design, the 2P mutation is quite
important. But that's orthogonal to this study because every sequence in our design do not have the 2P mutations. So in order to keep everything being equal, like control variable approach, we just have to to do everything without the 2P mutations in the original spike.
Awesome. And then I have a second question on your slide about linear design being effective independent of the UTRs.
Yeah, okay.
Do you believe that as the RNA gets shorter, as the coding sequence gets shorter, so the UTRs now represent a greater fraction of the entire mRNA sequence,
right.
and thus would contribute more to the overall folded energy of the MFE? Do you think this independence sort of decreases with decreasing RNA length?
That's a very good question. I think the independence, in other words, the code optimized baseline would have more problem probably with short sequences because, as you said, the UTRs actually correspond to a larger fraction of the whole sequence.
Yeah.
But our design, I think, it will be still mostly fine because we are so tightly structured that it doesn't have bandwidth to kind of have those kind of extramarital kind of affairs with the UTRs. So, in other words,
interesting.
Yeah.
So when you run the linear design, it doesn't actually take into when you run the actual fold part.
Right.
I imagine you use linear fold for
Yeah.
it. Does it actually fold with the UTRs, or does it just work on the coding sequence?
It just
during
works on
the
the
beam
coding
search?
sequence.
Okay, that's awesome.
Thank you.
Thanks so much.
Very much.
Thank you. Next question, Tatiana.
Yeah. So Tatiana Novobrantsova, Moderna. I wanted to ask maybe for a little more commentary, right, on how your increased double-stranded content, as well as the hypothesis of hidden uridines, work on the evasion of innate system recognition.
Yeah, right. sorry, that's just our hypothesis. I mean, I don't. I mean, we should have tested innate immune responses, but none of the reviewers suggested that, and we are not experts. So I mean, when this was published, I talked to other people, and they say, why don't you test kind of in vitro innate immune response? And it should be very easy to do in cell. I mean, I was like, I have no idea how to do it, but we really should to kind of redo the experiments and investigate why it just works so well without chemical modification. It's kind of still a mystery, and I think the uridium being mostly inaccessible in our structures is maybe one of the reasons, but I don't know, because Dr. Kariko's work suggests that uridium is important, like you should avoid uridiums because that triggers the toll-like receptors and other immune responses. We kind of embed it in structures so that it's probably not as accessible as the unstructured case. So that was our hypothesis.
And do you have any structural data from how those uridiums might be sort of formed in 3D structures?
Not 3D structure, just secondary structure. It's like either paired or unpaired. Yeah.
Thank you.
Thank you.
Okay, one more short question and short answer.
Sure. Thanks. Milad from Sanofi, very nice work. I just wanted to ask you about your opinion about the minimum free energy and the partition function, because you know that if you assume that the secondary structure of mRNA with that long length is a single graph, maybe it's a bit oversimplification of the problem. Right. I wanted to know if you, because you have also the linear partition work under, what's your opinion on, have you done any validation of your predictions with some experimental data? Thank you.
Thank you very much. That was a very good question, and thank you for referring to my work, linear fold and linear partition. And indeed, we believe that, just as this plot shows, that there are multiple alternative structures in the equilibrium. So it's never a single structure. There's a partition function over a Boltzmann ensemble of millions of different structures. Unfortunately, this work only optimized for MFE, minimum free energy. It doesn't really optimize for the partition function. But we believe that optimizing the partition function would probably be better. So we are actually working on it. To optimize the partition function, because you wanted the mRNAs to be tight but also flexible, also kind of plastic, so that when the ribosome goes through it, it can change shape as needed, right? You don't want the mRNA to be as hard as stone. That wouldn't work. You want it to be stable but flexible. Yeah, but that was some ongoing work. Yeah.
All right. Well, thanks again. Thanks for the
Thank you.
great discussion. Okay.